Um, it's an immense pleasure and honor to now welcome to Living Histories Sandra Govers from, uh, how should I say it, Sandra? KU.
From the K Leuven in Belgium, basically.
Thank you, Sandra, and turning it over to you to now tell us about Living Many Histories.
Okay. First of all, thank you, Sri, and the others for the invitation. It's really an honor to be part of this remarkable series. And I also enjoyed taking a little trip down memory lane and thinking about how all of these past events have influenced my own trajectory. And so one thing that has played a big role for me is what you're seeing here in this picture, because this is a picture of the city center of Leuven, where right in the middle you have its beautiful Gothic town hall. So this building was built in the 1400s. It's definitely worth a visit if you're ever in the area. But the reason that I'm showing you this picture is not only because it's beautiful, but also because, as you'll find out, much of my life has actually taken place, and it still does today, right in this city. Okay, but let's start perhaps with a brief introduction. So who am I now? Since 2022, I am an assistant professor at the Department of Biology of the KU Leuven in Belgium. So for those of you that might not be familiar with Belgium, Belgium is a small country in the north Midwest of Europe, and Leuven is located right here in the Dutch-speaking part of the country, which is highlighted in purple. There's also a French-speaking part of the country, which is highlighted in gray in this case. And then we also have some smaller German-speaking parts, but they are very small, and typically you don't find them on maps, just like this one. Then there at the end, you can see the building in which I'm currently located and where we do our research today. But more on that later. First, what I want to do is take you back to where it all began for me, which coincidentally or not so coincidentally was also in Leuven. And so for me, it started in 1998, sorry, 1988, right there at the Red Arrow. This is basically where I grew up in Leuven and where my parents still live to this day. And so my parents are Belgian, and they originally came from different parts of the country, but met while they were both studying in at university. They were both geography majors, and then afterwards, after their studies, basically just stuck around. My dad is now a geography professor at the KU Leuven here in Belgium, and then my mom first taught geography in high school, but then later on switched to ICT support and development for various companies. I am the oldest of their three children, so I have a younger sister and brother. And when I was young, I was mostly into sports, to be honest. So I played soccer, volleyball competitively. I was really into cycling as well. So despite my parents' background in science and academia, I wasn't particularly fascinated by it when I was younger. My dad did try to get me hooked on his rock collection. Didn't really work. We did have subscriptions to National Geographic and Scientific American magazines. I would read them occasionally, but not very often. My parents also once bought me like a small functioning microscope for my birthday, which I now think is super cool, but I don't think I actually spent much time using it back then. Um, but then after my very young years, I also went to high school in Leuven. It's a place called the St. Peter's College. It's located right there. So it's really not far from where I lived, which was mostly great because I could basically just roll out of bed and go to school. Then after high school, I chose to study bioengineering, also in Leuven, with the campus located right there at the bottom. And that eventually resulted in me doing a PhD in bioengineering as well. So right in that location. So at the very beginning, the mobility was extremely limited, but it was still very interesting nevertheless. But before I talk more about that, I feel that for me at least, for my trajectory, it's important to emphasize that for the majority of my high school and college years, I had absolutely no idea what I was going to do later on. And I think this rather ugly building that you can see here is a perfect example of this. And so I was a good student, definitely not exceptional, but one of the good things of being me in these years was that I didn't have to study too much to not fail any courses. And so a result of this was that I had quite a bit of free time available. So I used a large part of this to be relatively active in the bioengineering student organization. And so one year, still don't really know how this happened, but this actually led to me becoming the manager of the students' bar, which is the Knorrel, which is the building that you can see here. Uh, the bar is open from Sunday to Thursday, and as manager, you're basically responsible for a lot of things, for planning events, making sure you have enough volunteers to bartend, you have to order stuff, you have to negotiate with liquor companies, all of that kind of stuff. But also throughout the year that you are the manager, you live right there. So these three windows was basically the large room where I lived during that period. Um, as you can imagine, this was definitely not the healthiest period of my life, but it was one of the most interesting and educational ones for other reasons. Uh, Also interesting was that I lived there together with one other person, which was the accountant who was responsible for the bar's bookkeeping. And so he lived right there. So this other window was basically where his room was. And he also recently became a professor here in Leuven as well, which is funny. So there must have been at least something good in the water there, or at least a beer. But then for me, what was a pivotal moment was really the end of my studies. There was this moment where it all clicked, and that was during my master thesis in the lab of Kevin Verstrepen. It was a yeast systems biology lab where I was supervised by Aaron New. And so for those of you that might not be familiar with like how the system in mainland Europe works, master thesis, at least in Belgium, takes place during the final year of your studies, and it's where you basically become a full-time lab member and you work on a specific research project for an entire academic year. And in my case, this was looking into the yeast lag phase that cells go through when they switch from a preferred carbon source like glucose to another carbon source, non-preferred carbon source like maltose. And so what we tried to do was try to identify factors that affected the duration of this non-growing phase. And so this is really the period that for me, I started thinking that scientific research is really cool and something that I wanted to pursue. And so it's something that I really love and still love to do to this day is really the combination of thinking when you're designing and interpreting experiments with tinkering in the lab. And I think something that perfectly illustrates this are these little microscopy growth chambers that we use to construct ourselves. They were very rudimentary, but they worked, and you could generate these beautiful movies of yeast cells growing over time. And then we would actually go in and manually score them to determine when individual cells resume growth, which are the little arrows you can see here. After this period, I then joined the lab of Avram Aartse, also in Leuven, for my PhD. And so he basically convinced me to join his lab after a class on designing and using synthetic genetic circuits in bacteria. And I thought that was really cool. But then he also mentioned that he had a new microscope that his lab recently bought. And I think to me, that was all I needed to hear at the time to be convinced. And so during my PhD, what I did is mostly focused on microscopically characterizing different types of phenotypic heterogeneity in isogenic bacterial populations. And you can see some examples, some of the things we did here. And so a lot of what I did during my PhD was microscopy based, but it's also where I started to shift more and more gradually from manual analyses to becoming a bit more or making things a bit more automated and quantitative. Also, besides research, I was very fortunate to share this period with three great lab mates, William, Johannes, and Alexander, which at the time definitely helped me tremendously to navigate the highs and lows of scientific research. Then, after my PhD, I joined the lab of Christine Jacobs Wagner, who was then located at Yale University in New Haven. And so I had actually admired work of Christine's lab for years and was fortunate enough to receive financial support from something that is called the Belgian American Educational Foundation to go to her lab. And initially, I didn't really have a long-term goal point. I just wanted to spend a year there, learn more about cell biology, quantitative image analysis, and then go back to Belgium. That plan did not really work out that way, as I ended up staying for more than 3 years. This was mostly because it was such an inspiring, ambitious, but also in a collaborative environment. It was really a joy to be a part of. And a big part of that was Christine, but it was also all of the amazing people that she had there. And so during my time in Christine's lab is really when things turned very quantitative and I became interested in these very fundamental, basic aspects of bacterial proliferation. And so I had a main project that looked into E. coli cell cycle progression, but it was only It was actually during working on this project that we realized that some of my data was very complementary to data of a then graduate student in the lab with brain. And that turned into this collaboration that eventually led to this beautiful story on nucleotide scaling and how that affected the intercellular organization of translation in bacteria. But then, right around the time that Christine's lab moved to Stanford, is when I had to return to Belgium. But not all of my projects were actually finished. It was also a bit too early for me to start applying for faculty jobs. So I was in a bit of a pickle. And so at that point, it was actually Christine who reached out to Geraldine. Geraldine is a former postdoc of Christine that now runs her own lab in Brussels at UC Louvain. And she studies these remarkable predatory bacteria, which I've never worked with, but I'm just showing you a movie because I think they're beautiful. And Geraldine was really remarkable because she basically just allowed me to be a part of her lab while I was finishing my own projects, pursuing other grant applications, applying for faculty positions, and she basically just supported me and gave me feedback and advice without ever really asking for anything in return. And so I'm very happy that I'm able to actually show both of them on the same slide because I can confidently say that without these two amazing women, I would not be in science today. And so it's their support that eventually led to something that I still consider to be the best couple of weeks in science I've had so far, is basically where I got my position at KU Leuven in Belgium in 2022. And a few weeks later was awarded an ERC starting grant, which are these big European grants that really allow you to kickstart your own research group. And so now what we try to do in my own lab is really uncover these principles and mechanisms that underlie the subcellular organization of bacterial replication, where we try to move away from your typical model systems like E. coli, Bacillus subtilis, and start to explore this enormous diversity that you have in the bacterial domain. And so since the start in 2022, my lab has really developed into this great group of people that you can see here. They're lovely to work with. All of them are different, but they're inspirational, I think, in their own way. They're very kind, supportive, collaborative, and it really has been an absolute honor and pleasure to work with them. And so I hope to do this and continue this for the next 30 years or so. And then to end, I also want to emphasize the important role that these three people, two of them are still a bit tiny, have played throughout basically every aspect of my life. It's not has not always been easy to navigate life outside of science. Moving across continents was also not something that was straightforward. But my wife was always there to support me, and for that, I'm forever grateful. Our oldest son was also actually born in the States when we were there. And it's really to this day that all three of them are still the best support system I have, and also my favorite distraction from science and other things as well. Voilà. And so with that, I'm happy to thank you all for your attention.
Thank you so much, Sander, for a wonderfully inspiring talk. I'm clapping on behalf of the audience. We have some
Mhm.
time for questions. Audience, please send me your questions via chat. In the meantime, let me get started by asking you a question building off of something you emphasized in your talk, which is the role that mentorship has played in
Mhm.
in helping you develop into who you are as a scientist. Uh, how does this inform your own style of mentoring now that you're running a lab?
So what I try to do is be very actively aware of the important role you play or can play in these people's lives. Is that through these, well, sometimes even small gestures, you can help them feel supported and help them in pursuing their dreams. And so this is something that I myself now try to actively do and actively make time for each of them individually to find out what they want to achieve and how they want to achieve them and see where I can help and support them wherever I can.
Thank you. In the early part of your talk, you emphasized that you were really not following the obvious track of pursuing academic interests in your early years. You were actually playing a lot of sports and um, did not immediately take to your father's encouragement to pursue studying rocks. Is that
Mhm.
right? Um,
Mhm.
um, Now, how does that sense of play inform what you do in work?
I think the sense of play mostly comes from it's related to curiosity in my case as well. And so what I love most uh, of what we currently do in the lab is really this exploring other bacterial species. So we really have a bunch of them, and then my favorite thing to do is just throw them under a microscope and then stain a bunch of things so we can map them and really determine what is where, how big are the cells, where are the nucleoids, those kind of things. And so doing that with the new species is still, to me, the most fun part because we get to discover this completely new thing. And then, of course, just some more quantitative analyses on it. But I think that's the most playful part of the research we do today is exploring these new, well, they're not new bacterial species, but they're bacterial species that people typically have not put under a microscope.
Got it. Thank you again for a wonderfully inspiring talk and these sporting answers. In the interest of time, I'm closing the recording.